I think we're going to take our next speaker and get started with that. And then, Derek, I think you'll be able to hear us and should be able to hear some questions as we get into a bit of discussion at the end here, if you're agreeable to that approach.
Yes, that's fine. Okay.
So
I'll turn my microphone off so I don't confuse things, all right?
Our next speaker is Cote from EPA.
Thank you. I really appreciate that talk, Derek, that you just gave. So I work for the Environmental Protection Agency in the National Center for Environmental Assessment. The National Center for Environmental Assessment's mission is to evaluate health and environmental effects data as it speaks to public health risks, in particular focusing on hazard identification and dose response assessment. And as I had alluded to in my opening remarks, a number of years ago, I was asked to begin to look at how we could better integrate molecular and systems biology data into risk assessment. So that's what this talk will be about. And in particular, I hope to highlight how I think we may transition from where we are today to where we want to be in the future, and to do this balancing act between moving too quickly versus moving too slowly, and how we can incorporate our current state of knowledge, clearly understanding that in 5 years, 10 years, 20 years, it's going to be significantly different than it is today. So this is the outline for my talk. So in this effort, one of the things we decided to do was to, I think the goals of what we want to accomplish are pretty clear to everyone and have been much discussed. And I think why this kind of effort is important is also pretty obvious to everyone. The way we chose to approach this problem, as Kim had alluded to, is through the development of specific prototypes. It's very difficult often for people to go from the very abstract strategic discussions of these issues to the practical application without something in front of them to work with. And so the purpose of these prototypes are to sort of engender a broader discussion among the risk assessment community about how we proceed. And in particular, we're interested in what are the decision rules that you need to apply to emerging data that gets you to the place, sort of gets you to the right answer, if you were, and how to incorporate these advances into risk assessment. So the basic approach that we're using is we're trying to take we chose prototypes that had both existing molecular systems biology data and as well as traditional data, and we're trying to reverse engineer between the two and figure out what kinds of new approaches that we need to apply to this emerging data, again, to kind of come up with what are credible answers. I think without these sort of proof of concept examples that the wider risk assessment and risk management community will be leery of proceeding. So the other thing that we're trying to do, although we're really looking at this in terms of how the National Center for Environmental Assessment is going to begin to alter risk assessment, what we're trying to do is maintain a dialogue with other federal agencies so that we can benefit from their thinking and advice on this topic. So if we talk about risk context and approaches, if many of you will remember from Science and Decisions, one of the key pieces of advice is that not all risk assessments needed to do everything, and that the risk assessment that you develop should be targeted to the kind of risk management problem that you're trying to solve, or the risk context. And so I'll briefly go through how we thought about this problem. In reality, when you start thinking about the kinds of environmental problems you want to address, it's largely driven by the number of chemicals that you have to deal with at a time. And we've separated our thinking into these three broad categories, which are really on a continuum. They're not distinctly separate. So we started out with thinking about the kinds of problems that one has to deal with depending on the number of chemicals you have. And the increasing need of confidence as you move from the vast number of chemicals to a smaller number of chemicals. So these are the kinds of environmental problems that we essentially came up with. That wasn't good. There. So on this side, sort of just screening and ranking, which has been referred to when you're dealing with large numbers of chemicals. Limited decision-making here in the middle, where you might be looking at 1,000 chemicals. And when I talk about limited decision-making in this context, generally what I mean are, in general, but not exclusively, non-regulatory decisions. The agency makes lots of non-regulatory decisions. An example might be at a Superfund site, a specific site where you're trying to decide what dirt to move and how much dirt to move, would be an example of an important decision which was a non-regulatory decision. But also a decision in which you might be able to tolerate more uncertainty than when you're over here at the under the Tier 3 column, which are, I often refer to the everything that can be done will be done chemicals. And here we're talking about a more limited number of chemicals, but things generally that are characterized by national exposure and regulatory decision-making. So then if you start matching up, what kind of data can you bring to bear to answer these questions? If you're talking 10,000 chemicals, you're talking high throughput only. I mean, that's just got to be true. If you're talking about thousands of chemicals, you may have the opportunity to bring in data that takes you a bit more time to collect and analyze, but may be slightly more robust. And then lastly, in the Tier 3, I think you're looking at environmental exposures and the most realistic scenarios, and increasing level of evidence. So here are just some when I think of these kinds of things, I think the QSAR, the high throughput ToxCast kind of systems are what you're talking about over on this side. When you begin to add in, and this builds, as Kim had referred to, these are not separate, but one building on another. You can introduce short-term exposure test systems using either mammalian species or alternative species, tissue constructs like tissue culture and perhaps in silico modeling. One of the things that you get is improved metabolism. One of the drawbacks of these systems at this point in time is that their metabolic capabilities are not always very robust. Again, you're adding in different types of assays, and you're also beginning to look at traditional data. And then when on this side, we're talking about the most robust of kind of test systems that also take the longest. Often, though, however, looking at environmental exposure. And when you're talking about risk assessment, here you're considering sort of all policy relevant data, because the more limited number of chemicals allows you more, the luxury of more in-depth analysis and more time. So when we're talking about hazard identification, and this is like trying to figure out if chemicals cause a specific health outcome, I want to just, what I'm going to do now is review some of the basic steps and then go on to show you some specific examples. So, I think that we'll have to have explicit inclusion and exclusion criteria. The reality is a lot of the studies we've done today, I think you look back on them and we wish we would have done them differently. So not all data is sort of created equal. What we're looking for are molecular patterns of events that we think will cause a chemical to be more likely to cause an effect versus another. Two points underneath that phenotypic outcomes can depend both on tissue and organism level integration, and as Lauren pointed out, life stage, tissue and species can make a big difference in terms of phenotypic outcome. You may have very similar molecular events with different phenotypic outcomes depending on some of these features. In general, we think that we need to be looking at the pathway or network level. That knowledge of single events or single genes is not sufficient in terms of true understanding. And I wanted to make a point of at least EPA, and I think the Europeans at this point, when we talk about adverse outcome pathways versus mode of action, that we're considering the adverse outcome pathway considers population-level dose response, where mode of action might be more at the individual. So I think when you move from individual dose responses to population dose responses, there are some additional issues that need consideration. You know, the traditional Bradford Hill criteria we've found very useful. You know, we want to look for things that are plausible, coherent, consistent across studies. And then I think where we're moving is to define new types of effects that we will call adverse in themselves in the absence of phenotypic data. Now I want to turn my attention briefly to dose response, which is an even trickier issue, I think, than the hazard identification, which is complicated enough. And right now in the various prototypes, there's a range of approaches that are being used in the prototypes. And we're going to look at all of them, and they have pluses and minuses in each case. And I'm going to lay these out in sort of increasing complexity. The simplest case would be to, for those of you that don't, you know, spend your days thinking about risk assessment, the lowest effect level, the lowest adverse effect level, the benchmark dose, which is essentially a lowest effect level established by modeling. So you could essentially, if this is a collection of dose responses from new types of test systems, omic data, it could be proteomic data, it could be a variety of things. But one could essentially just build on the current traditional approach of identifying low levels of effects and dividing by uncertainty factors. I find this an intrinsically not terribly satisfying way to proceed, but perhaps the easiest for some risk assessors to understand because it's the closest to what we currently do now. The next level of complexity would be to consider the slope of the dose response within the experimental range. I'm not talking here about low-dose extrapolation, but again, to consider the full range of exposures, preferably at environmental concentrations. And you could use essentially the slopes of these lines to compare the potency of chemicals. The other complicating factor as you build in complexity is that you may have multiple assays that interrogate the same network. And so how do you integrate this information? One of the things we know, it's not just the potency of a chemical that predicts its toxicity, but as well as what is called its promiscuity. How many different pathways does it disrupt? So this is David Reif's work from the National Center for Computational Toxicology. What he's done is, in this particular case, color-coded different kinds of assays that interrogate the same system. The size of the piece of pie represents the potency in that particular assay. And then displaying them in kind of these grouped ways so you get a sense of both the potency of the chemical in various assays as well as the how many different pathways a chemical might disrupt, how many assays it's positive in. I keep doing that. So if you go to the next level, we've begun to describe essentially wiring diagrams for parts of networks, not the entire network. But already, this is a slide I borrowed from the alternative species folks. This is alternative species data. And you see in the dose response, it's a fairly complicated dose response, which they can explain by understanding the sort of the wiring diagram for the biologic processes and the feedback loops. I got this slide from Ed Perkins, and our Army Corps of Engineers, which is doing a lot of our alternative species work and has been a good collaborator. And lastly, this is kind of where we're going, where we want to be in some ways. Where we have a much more complex description of the network of biologic events that lead to disease. I think it's probably almost emblematic that MIT is working on a wiring diagram for cancer. They're kind of well down that road of trying to lay that out. But just having this alone is insufficient in itself. As Lyle Burgoon, who works for EPA, often says to me that this is essentially a roadmap of disease, but what you're really interested in is the traffic flow on that roadmap. You're really interested in traffic patterns. So people are applying the kinds of systems approaches that you use in understanding computer network flows, as well as power grids. A lot of other kinds of modeling work is being brought to bear. And lastly, I want to make the point that there's really no reason to treat cancer and non-cancer differently when you're talking about these underlying biologic processes. And in fact, when you're looking at the molecular level, these biologic processes don't parse easily into cancer and non-cancer. So I think if we try and set up a different way to handle them, we're essentially just going to, or kind of asking for trouble, as it were. So other steps that are really important is this issue of what's the dose? What's the exposure-dose relationship? Is become much more complicated. And here I've just listed several approaches in the prototypes that are being used. Reverse dosimetry modeling in the in vitro exposures, starting with the concentration that you're exposing cells to and trying to model back to what the environmental exposures would be. Monitored exposures and conventional dosimetry using biomarkers of effect and exposure. So those are all approaches that can be taken. Probably not a comprehensive list. One of the things about a lot of the in vitro studies that you can now use human cell lines. So to some extent you address this species extrapolation issue, but it's going to be we have a lot of animal data. We're going to continue to use animal data for the foreseeable future. And I think to be able to understand how the species relevance of the data that you're looking at is incredibly important. It's been mentioned several times, characterizing variability among humans, and this issue of background response. What's the already existing pre-exposure background biologic state of the individual, as well as this process of adaptation, and how you incorporate both of those into your population models is going to be a key issue. So again, as I mentioned, we're interested in population risk, including variability and uncertainty. And the goal, I think, as it's been for many years with all kind of risk assessment, but it's particularly on people's minds now, is to begin to replace with data and our extrapolation and uncertainty factors. And a clearest example for me is we now can essentially do dose response assessment at environmental concentrations. So we're really able now to push down the dose response range that we look at, perhaps obviating the need in the near future for low dose extrapolation. So next I'm going to go through some specific examples, and those specific examples are going to be tied to both the risk context tiers that I mentioned earlier, as well as I had indicated some of the types of data that you might look at in each of these tiers. So this is one of many slides that I've stolen from my colleagues at the National Center for Computational Toxicology. And they have been working for 8 to 10 years now on developing specific assays that interrogate different parts of the biologic processes. And they have a whole collection of assays at this point in time. And they do a lot of work looking at the validity of these assays, the specificity of these assays. And from this large set of assays, this subset of, I think, 150 assays that are being used in the TOX-21 program have been drawn. And for those of you who may not be familiar with TOX-21, it's a collaborative effort among several federal agencies to test 10,000 chemicals over the next 2.5 years. So that kind of large-scale effort really puts the onus on what risk assessors are going to do with that kinds of data. But the assays have been drawn from this subset, from this larger set of assays. One of the things we also know, in spite of how many assays we have that successfully interrogate important biologic processes, we currently, it's still insufficient. There are a lot of processes, chemical events that we still miss. So, the false positive rate is considerably better than the false negative rate at this point in time. But again, I think the point is that we can successfully interrogate many biologic processes at this point in time. So in using these kinds of data, what I would propose, since if you have a set battery of assays and data likes coming out of TOX-21, what you'll have is consistent testing in a battery of assays for 10,000 different chemicals. And so one of the things you can do is to look at the relative potency of these chemicals in these assays and rank based on that. And you can benchmark these chemicals to chemicals that we know a lot about in terms of anchoring the results of these assays. If you have the exposure data, you can also readjust the ranking, essentially looking at margins of exposure. And there's also, we're beginning to incorporate population variability. The amount of population variability data we have is to date scant. But I think everyone recognizes the importance of it, and so we're trying to build this framework where it's a blank space to be filled, and it encourages people to fill that space. And there are several very robust approaches that are being taken to collecting larger amounts of population data that we don't really have time to go into, but the last NAS workshop, of course, was strictly focused on population variability. So when you consider these factors, the goal would just be to put chemicals into broad categories of high, medium, and low. And my expectation, it would be the chemicals up here at the high end and those with no data that we would then begin to focus our attention on. And then I think if you take the chemicals that rank high in terms of both their promiscuity and their potency, you can then begin to sort them into mode of action categories based on these various assessments. One of the important functions of that is both beginning to zero in on hazard identification, but as well to structure targeted testing for additional data gathering. Lauren showed a different version of this slide, but one of the really difficult issues in this Tier 1 level is how you integrate traditional data with this high throughput data where you'll have the same battery of assays consistently applied. And, because they're Ames assays, there might be an LD50. I mean, you don't want to ignore that information. But one of the really tricky issues at this point in time is how you integrate QSAR information, how you integrate high throughput, as well as traditional data, but still one is able to do it in a high throughput fashion. So that's work in progress that I think is going to be critically important. So if we go to this sort of next level of assessment, and here we're using test systems that are more metabolically competent, you have more integrated, you can get tissue integration information as well as in some cases organism level of integration. And the aim is to, here in this case, the aim is to generate a reference value versus the more cruder binning. I'm still not convinced that the best application of these data is still not a relative ranking of potency. But the goal would be to generate some kind of a reference value. And as I've alluded to, the hallmarks of some of these assessments are more intact metabolism, higher level of biologic integration, additional endpoints, like particularly looking at the alternative species. You can begin to look at things like neurobehavioral endpoints that are difficult to look at in strictly in vitro assays. And the good news and the bad news is this last point. Often what people are looking at are omics at perhaps the tissue level. And what you do is you sum together large numbers of different cell types, or at least a variety of different cell types that may each have their own omics. And so sometimes figuring out what's going on in these different cell types is important and more difficult in these kinds of test systems. So I'm going to show you two examples. This is Rusty Thomas's data. Rusty's been working for a good while now on examining if you can look at short-term exposures, look at initiating events, and predict health outcomes that might occur substantially later in the disease process. And so he's been looking at, these are lengths of exposure, and what you see is that Rusty gets really quite good correlation between the in vivo outcomes and the in vitro outcomes. I think this is the cancer data. He also has data for non-cancer endpoints. So what this would argue that from the transcriptomic data, you can at least get some sense of the relative potency of chemicals to induce longer-term events. This note here is one of the tricky issues is, of course, at this point in time, you're looking at reams of dose-response data from various genes, individual genes, and how do how do you kind of integrate that in meaningful ways? And Rusty's chosen to look at the benchmark dose approach for each gene and then average across the pathway. This is another example from the alternative species community, where what you see in red is sort of the comparison across where you have traditional and can look in vitro in an alternative species, and the green is areas that you get results from the alternative species and more insights into chemicals that you did not get strictly from the in vitro data. So a little more robust, we think primarily because of the metabolism issues, that you have better metabolism in alternative species. But these are other kinds of data that one could use in a Tier 2 assessment. And then lastly, I'm going to talk about the Tier 3, which, as you remember, is kind of the everything that can be done will be done chemicals. And I frequently get the question, why are you bothering to look at these chemicals? Because we already know so much about them. We have rich, in vitro database, traditional in vitro databases. We have epidemiology, we have clinical data, we have animal tox. Why in the heck are you focusing on these? And primarily, we went into it looking, wanting to explore the proof of concept, looking at how predictive are these in vitro approaches of public health risks, which we're truly interested in, and how do you make those relationships? We're looking at informing difficult issues that are not well resolved by traditional data. I'd mentioned low-dose extrapolation earlier. Population variability is another one. And then lastly, extending what is learned with chemicals of less data. If you can characterize a disease process that is set in progress by a chemical, then if you see that same pattern of molecular events in chemicals that you don't know much about, I think it's reasonable to say you're concerned about those. And I'll give a couple of examples. So, to focus on what I'm going to just briefly touch on are the prototypes that we have spoken of, that we've been focusing on, ozone, benzene, and PAHs. Waiting in the wings are several others, some of which Kim alluded to earlier. But we're looking at causal associations between exposure and traditional upstream events and phenotypic outcomes. And people have a better handle in these case studies on this issue of multiple cell types. It's not completely resolved, but people have been giving that issue some thought. So just in terms of this issue of pattern recognition, I pulled together several of the knowledge maps that have come out of these efforts. And I think I just want to make the point that what you see are specific patterns that are associated with specific diseases. And I think the evidence for the causal relationship is strong in these cases. So it just provides us an opportunity to both look in depth at a disease process as well as extrapolate to chemicals that we know less about. And Bob Devlin's going to talk about this specific example later in the day. There's a lovely set of papers by the Berkeley group on benzene. And I'd have to say the PAH one is still kind of a work in progress. So I think that what we can say, though, from these prototypic examples that are either have been developed or under development, that you can see specific alterations in gene expression profiles that are consistent, coherent, and biologically plausible indicators of both traditional upstream events as well as phenotypic outcomes. They're both dose and time related. The time issue, which has been alluded to, is a particularly sort of complexifying factor, but to borrow a phrase from Peter Proust. But you can demonstrate experimentally the relationship between these upstream events, intermediate events, and phenotypic outcomes. So it one of the things that the relevance, sort of difficult issues that are kind of waiting in the wings, are, for example, with the benzene example, we have this lovely robust data set in humans. NTP's doing an extensive study on animals and benzene and looking at molecular patterns. So it'll be fascinating to compare those two data sets. Human susceptibility we're working on. I think Bob will probably have one of the best examples of being able to explore human variability using these. I hope I'm right, Bob. But I think what it argues that data with limited chemicals with the same signatures are certainly at least of concern for further evaluation. So I think better understanding these disease fingerprints can really help us in terms of the Tier 1 and Tier 2 kinds of assessments. The other thing that I think is important here in terms of how we transition from where we've been to where we are is I think rolling out these kind of more molecular-based assessments integrated with our traditional look at risk assessment. And I think what we can do is to begin to make people more comfortable with the new types of data. We can explore better exactly what our decision rules are for using that new type of data in risk assessment. And then I think gradually you move from the old model to the new model. So what I envision in the near future is seeing assessment of molecular biology data done in tandem with traditional data, and that those are presented together. The external peer review system has an opportunity to look at those assessments sort of in tandem, and thereby we would sort of move forward in that process. So in summary, I think hazard identification and dose response assessment are about pattern recognition. I can't emphasize this enough, and it becomes particularly difficult when you have reams of new data. How do you find the correct patterns? One of the debates we have with the Tier 3 risk assessments frequently are, if we didn't know in advance what the outcome was, could we have found it? It's clearly there when you know what to look for, but could we have found it in the absence of the traditional data? And there's kind of an active discussion about that internally. But I think when you identify these patterns at the pathway and network level, you can use them both for hazard identification and estimating potency, and characterizing a variety of uncertainties, informing those uncertainties, reducing those uncertainties. One of the things that was a shocker that I wanted to make a point of is in terms of the number of chemicals with adequate data is still very limited. When we went out looking for prototypes where we had really very high criteria for the kinds of data we wanted, there were not many chemicals that we had sufficient data on. So I think going through this risk assessment process, identifying what kind of data works, what kind of data we wished we had, will also better inform the research community and will result in the production of better data. The weight of evidence, as I alluded to in these different tiers, is different depending on the type of problem that you're trying to solve. And I think that it allows you to use different kinds of data to inform different types of situations. And then the last point is what everybody's, I think, already said, is that significant progress is amazing progress is being made. But more needs to be done, and I think the trick will be to utilize as best we can the data that we have now, recognizing that our perspective of the world is going to be quite different in 5 and 10 years. So, what you'd like to do is move forward without shooting yourself in the foot. I think Kim had alluded to not making any serious mistakes. Lastly, there's a whole bunch of people that we've worked with over the last few years, and here's a partial list. I erred in leaving Derek Knight off this list, but he should have clearly been on here also. So these are people I'd like to thank. So thank you.